We're moving on to talk about how we can actually use the information that we have about DNA to possibly solve some problems. So we talked about crime scenes and paternity cases, but now we want to talk about actually using the genes that we found. So first we have to find the gene. And that's what the DNA fingerprint can help us do. DNA fingerprinting can help us find certain genes. DNA fingerprinting can help us find certain genes. Once we found the gene, we can actually add the gene to a new organism. And so genetic engineering, we want to add in the word, it's the purposeful modification. Because remember, your DNA can be changed by mutations, but purposefully modifying DNA is also called creating recombinant DNA. Now this word recombinant sounds like recombined. Recombined, recombinant, meaning the same thing. We're combining two different sources of DNA. So in this picture, this is a little primate called a marmoset. And the marmoset has in its paws a glow gene. Now this glow gene is normally only found in fireflies and some glowing jellyfish. So why would they put the glow gene in this monkey? Well, it's not just so that the monkey glows in the dark. It's actually they put the glow gene and a new gene, like for example the gene for insulin. Think of all the people who are diabetic, who if we could give them the insulin gene, they wouldn't be diabetic anymore. However, how do we know that it has picked up the insulin gene? And so what they do is they take the glow gene, they put it next to the other gene that we want to put in there. And if both of them go in, then we see it glowing and we know the other gene made it in there as well. And so a transgenic organism contains foreign DNA from another species. It's where we've taken DNA that doesn't normally belong there and we've put it into that organism. So trans means between. So it's sharing DNA between organisms. And the way that we do that is we make this new DNA. Recombinant DNA is artificial DNA that is created by combining two different sources of DNA. So it's not like genetic engineering is going to give humans wings or gills. That's not why we're looking at genetic engineering. Genetic engineering is mostly being looked at to cure genetic diseases. But genetic engineering is really difficult because we can't just go in there and do surgery on your DNA. It's a lot more complicated. So we see that we've been really successful, science has been really successful at genetically engineering bacteria. The most common organism that we genetically engineer is bacteria. And this should make sense. Remember, bacteria are just a single cell. They are they're prokaryotic, so they don't have a nucleus protecting their DNA, so it's easy to access their DNA. So bacteria, this picture here of this bacteria, these are the ones that we've been most successful at genetically engineering. So we have a couple steps here as well. The first step is that the restriction enzyme is used to cleave. Remember, cleave means cut, like a meat cleaver is a knife that cuts. Restriction enzyme is used to cleave the desired gene from a DNA sequence. For example, insulin. One of the most famous examples of genetic engineering is with human insulin. Insulin is found only in mammals. And so it used to be if you wanted insulin, we would take the pancreas of a cow or a pig, grind it up, isolate the insulin, and that's how diabetics got their insulin. What we've been able to do now is actually put the insulin gene into a bacteria, and the bacteria will make our insulin for us. So the first step is to find the gene that we want and to cut it with the restriction enzyme. The same restriction enzyme is used to cleave the vector. Now the vector is the structure used to carry the foreign DNA. It's the delivery vehicle. delivery into the cell. And bacteria plasmids are really commonly used because they are circular DNA found inside of a bacteria. So remember, we're most successful at genetically engineering bacteria. We cut out the gene that we want, for example, insulin from a human. We cut open the plasmid. This is going to be our delivery vehicle. This is how we put the gene we want into the vector. And then we have to put the vector into the host cell. So the foreign DNA and vector are spliced together. Splice means to combine. The recombinant DNA is inserted into the host. Remember, bacteria are who we're successful at this with. So we take the isolated gene that we cut with the restriction enzyme, the plasmid that we opened with the restriction enzyme, and we bind them together. And this is our recombinant DNA molecule. And so this is the same picture that you have in your notes. The recombinant DNA is now inserted into the host, and the host will copy and produce the protein. So because it has the gene, it will actually create the protein, even though it's a human protein. Insulin can be made in really large quantities in this way. And we want to end by looking at this particular example here that looks at, here's our donor gene coming from a human cell. Here is our bacteria plasmid. We do want to note that we use the restriction enzyme to cut both of them. Cut both of them. Cut both. And then we splice them together, and now this is our recombinant DNA molecule. We put the DNA into the bacteria, and this would be called a transgenic organism, because this bacteria now has DNA that doesn't normally belong to it. And so these are the steps for genetic engineering. This is how we get insulin, human growth hormone, and we're hoping that we can make other hormones this way as well. However, we do have some concerns. Should we be making artificial proteins? Now, if you have someone in your family who's really sick, you would give anything for them to be healthy, and so genetic engineering might be the right decision. However, we are worried sometimes about allergic reactions. If we add a peanut gene to a corn plant, does that mean that someone will now have an allergic reaction to it? Stem cell research is used on a lot of these experiments with transgenic organisms, and some people aren't sure about whether we should be using stem cells. There are also environmental problems, creating superweeds, antibiotic-resistant bacteria, and destruction of beneficial insects like bees by pesticides. So remember, we have a lot of questions about should we be doing these things. And so we do want to spend some time thinking about, well, it's great that science can do this, but is it the right thing for us to do? This is where we'll stop our notes for today.